Howdy, howdy, howdy.
Hey guys.
Randy, how
Hello.
are you?
I'm doing well.
Good, good.
Awesome. Awesome.
Yeah, we already have a crowd tonight.
Wow.
100 and some people, and we're not even live for but a couple minutes. That's good. That's good.
Where are you at?
So,
Where are you located?
huh?
Where are you located?
So I am in Northeast Ohio,
Mhm.
in the land of just never-ending winter this year.
Okay.
Our winter this year was pretty darn harsh. Um, and I know you would probably appreciate this being as scientific as what you are. I did the old experiment where you walk outside with the warm water, and I threw it in the air, and it made snow before it hit the ground.
Oh, wow.
Yeah,
Impressive.
It's pretty cold.
we
Yeah.
were. I think we were like minus 22 that
Oh,
day.
geez. I was up in Alberta, Canada, last month, and stepped outside. Said, "Wow, I can see my breath. I haven't seen my breath in a while here in California."
Yeah.
Yeah. Yeah. I'm located in southeast Alabama, so we made splits the last 2 weeks.
Okay. All right. So
So
we're an almond pollination right now, getting towards the end of bloom. So um, yeah, a little bit earlier season, similar to yours. Although we're not ready for splits yet.
yeah.
Yeah.
Well, I did. I came across some queens from Zach Heinzelman in Hawaii, and
Okay.
I've been my bees down here. My local Alabama bees are a little spicy, and so I'm trying to calm them down. In large part, honestly, I heard your interview with Brian a few months back, and you talked about how that is an extremely important
For me, yeah.
trait for you. And I've heard many other people say that. And I've never really enjoyed. I thought it was kind of okay to have defensive bees and work them, but I'm kind of the point now to where after I've seen some of the nicer bees, I'm moving in that direction. So that's kind of becoming my number one trait. Of course, second is productivity, and then third is going to be mites and mite control. And so I'm kind of at that stage where
That's our order also.
kind of switching my priority a little bit. It was production, but now I'm just humbling the spicy ones. And I made a bunch of splits. I tried this last year with some of Zach's queens, and it worked out really well. And that was about half of my operation. I've got pretty calm, and now I'm working on the other half that they like to staple your clothes to your body,
Oh my gosh.
and I'm trying to get those calmed down too. So we'll see how that goes. It was
So,
perfect weather, mid 60s a couple of weeks ago. And this past weekend it was a little chilly, but I think we're gonna be okay.
okay. I was in Serbia and Bosnia last month and visited a place where they had like a bee nature center. And in that center, they had a room or two rooms for apitherapy, not the stinging apitherapy. One, you had a flat bed, plywood bed with ventilation, and there was a beehive underneath you. You lay down, and
Oh,
you got
wow.
the vibe of the bees and the odor.
Mhm.
The other was beehives with a tube coming out and a little fan, and you would put a mask on and inhale the odors of the hive.
Hmm.
The point that we're making is that part of the enjoyment of beekeeping is to enjoy the vibe of the bees.
Mhm.
When the bees are calm and have that calm smell, it's
Mhm.
very it's a vibe that all beekeepers appreciate. And this is something that I hear from the people who work with veterans with post-traumatic stress syndrome, with people in prisons, that with the veterans, they go from a very high incident rate of suicide to zero. Something about just that vibe of the beehive, that pleasant feeling. In my beginners classes, back I've been teaching beginners classes for well over 50 years. I used to go through all the how to set up and the assembly equipment and all the facts and everything. And now all those facts are out there on the internet. So what I now focus on is learning how to handle bees.
Mhm.
And by the end of or halfway through my class, after some instruction on handling bees with a PowerPoint presentation, we go out to a bunch of nukes. And within a half an hour, nobody in the beginner class has gloves on, and most have veils off, and are happily handling bees and just enjoying that vibe of the bees. So it's a whole different thing of approaching them from an adversarial, protective standpoint versus approaching them with just calmness and relaxation and
Mhm.
cooperativity.
Yeah.
That's great. I'm not quite to that point yet. That's kind of my goal, and I don't mind wearing a veil, but I'd like to get to the point where I didn't always have to wear gloves and a
Oh
jacket.
yeah,
And
yeah,
so and if I'm not in a hurry, if I'm just, I have limited time. I do work full time. I'm a sideliner beekeeper, and so I just kind of got to get it done in a couple of days every week. I run a little over 100 colonies, and so sometimes I have to move a little quicker than I would like. But when I can just take my time, and the goal is to be when I can just take my time and learn how to be more efficient, that I can get to that point to where I don't have to wear gloves. I don't always have to now, but I've always got everything available in case I need it.
yeah.
But yeah, that's my goal is at least to get them toned down to where it's just, just that vibe of the bees kind of working across the frames, that kind of waving, kind of moving
Yep.
gently across the frames, not too worried about you. That's what I'm looking for. And I've got quite a few that are that way now, but I've got quite a few that aren't. So we're
Right.
working on that.
It's worth
But,
it.
Randy, I think tonight, the topic, a lot of people are concerned about this the colony loss, the losses that they're having. I know commercial beekeepers, a lot of them are having tremendous loss, and a lot of hobbyists too. Brian's had some struggles. A lot of people have, and I think kind of what we'd like to focus on a little bit tonight, if we could, and is what are your thoughts on that? Have you seen losses out there more than usual, and what are your thoughts about what's going on?
Oh yeah, we definitely got hit in our operation. This is the first year I can think of that we came up short with bees for almond pollution for our contracts. We normally go in at above top strength with prime contracts year after year. Let me ask Brian first. What field signs, did you see in your bees? What did you observe?
So for my colonies, the last two colonies that I found that were dead outs, they were, I just was out and I filmed one today. They were, at this point in the year, they were over 4 frames.
Uh,
So it was, for this time of the year here, decent enough of a size cluster.
yeah,
I had insulated inner covers, and when I looked in the insulated inner, the little hole I have my Hive Alive fondant above that, I looked down in there and I could just tell they were dead. I saw, and it was funny because when I pulled the insulated inner off, the whole cluster was dead, and even there was like a small little portion where you could tell like they died, almost like they just flash died, even up the little tube that goes into the insulated inner.
wow.
And I when I saw that, I was just, well, that is really weird. And frames, like there were literally bees that were dead hanging on the frame. I had very minimal on the bottom board.
Uh,
And the bees that were on the frames, I mean, they were dead literally 2 or 3 cells over was capped over, honey.
okay,
So they're so close to the food. They died literally not even a centimeter away. Like the top edge was not even a centimeter away from the fondant that was there. So they were right
yeah.
next to the food.
Did they die with their heads in the cells, or
Some of them were, some of them had their heads in the cells, but for the most part, like, there was a vast majority of them that it's like they were just hanging on the frame.
okay?
And I'm just looking at it like, well, what gives?
Yeah, that's very interesting. Okay, that's not what we're seeing with ours. What we're seeing is in the fall failure to respond to feeding a pollen sub and syrup. We in California we don't have a pollen or nectar flow in the late summer fall.
Mm-hmm.
So starting September, we ramp them up by feeding pollen sub and syrup
Mm-hmm.
continually and build them up big to go into the winter.
Mm-hmm.
Some of the colonies in the yards responded normally. Other ones, instead of growing, they went downhill, just dwindled away. We didn't see any dead bees at all. Um, so it was the adults would obviously fly out, and the and they all were rearing brood. But this is similar to what happened with CCD when Nosema ceranae invaded. The typical sign of a Nosema ceranae invasion is that once the infestation rate gets very high, the adult bees start foraging earlier in life. So they go from nursing right to foraging, and once they start flying, they they die quickly in a few days, or they commit altruistic self-removal. If they're feeling sick, they just fly out and don't come back.
Hmm.
So what happened? You see colonies with brood, but they couldn't build because the adult workforce kept decreasing, and they couldn't cover any more brood, and you wind up with a silver dollar patch of brood with a queen, and all the rest of the bees gone. Now, if you dissected those bees, 8 out of 10 or 10 out of 10 would be infected really bad with Nosema ceranae. The
Yeah.
other thing is there were viruses involved back
Yeah.
then. Question about which viruses. I was involved with a startup out of Israel, and we actually created CCD in the yard by inoculating them with a virus on top of the Nosema ceranae, and we saw CCD at every stage of its progression. So that was very interesting seeing that. So my first suspect, of course, was Nosema. But I went out to our hives, took a bunch of samples, and it's not Nosema. We can find Nosema occasionally, but it was not associated with the collapsing or the strong colonies. So we're kind of a control group. My own personal operation. We're not in an agriculture area, so we can rule out that it was pesticide exposure. We haven't used a synthetic miticide for over 20 years, so it's not miticide buildup in the combs.
Hmm.
We had varroa very well under control, so we know
Hmm.
it wasn't unmanaged varroa. We were feeding them, so we know it wasn't lack of food. So it gets down, for me at least, unusual weather conditions. We were hot and dry, but we've been hot and dry before in the
Hmm.
fall. The thing that makes me think it's not environmental is that it didn't affect all the hives in each yard. We run 55 yards of bees, so we have a bunch of yards to compare.
Hmm.
And my boys are running controlled trials testing three pollen subs for a manufacturer. So they were getting documented continual feeding of these pollen subs. Okay,
Right.
but you have a colony that would respond, and the colony right next to it that would just dwindle down to 2 frames of bees.
Hmm.
That makes me strongly suspect there's a pathogen involved. I strongly suspect that it's either a novel virus or a variant of one of the existing viruses. I'm currently working with a USDA researcher, been taking samples into him. He's doing some metagenomic analysis and independent of the other inspectors or researchers. So the more people looking at it, the better. On the 28th of this month, Project Apis M has an open meeting that will review everything that the USDA has learned up to this point. What I'm seeing out my sons just came back from almond pollination this afternoon. They went down to pick out some breeder colonies. Now we have a bunch of breeder colonies that are mite free, never haven't been treated in a year, and they're looking great. They're big and strong and looking fine.
Some.
So I haven't clarified whether they across the board had fewer issues than the other
Hmm.
ones, but they brought back a truckload of about 20 of the breeders that had documented mite counts of 0 for the entire last year.
Hmm.
And then they brought back some other strong colonies, but they said to find another 20 strong colonies to bring back that were not breeders, just to make the self starters, they had to go through 200 hives. Normally, we just go down and pick up the first 20 hives, and they would all be super strong.
Yeah.
So they had boosted everybody up to 8 frames 2 weeks ago as bloom was starting, and some of them actually decreased in strength since then. So I
Wow.
was hoping that we were going to be seeing everybody recovering, because as we've been looking at them, they've
Hmm.
been really brewed very nicely in the almonds. They got very good nutrition coming in, lots of brood going, and but the question was, the brood doesn't mean anything. When those bees emerge from the brood, will they stay in the hive and then survive enough to expand the brood nest and give the queen more room to lay? If those bees emerge and then fly out early, then the understand that this time of year, you've got, if a queen is laying 1,500 eggs a day and the nurses are feeding them, you'd have a recruitment rate of
Hmm.
1,500 eggs a day, new bees a day. At that rate, A colony during normal buildup increases by a net of about 500 bees per day. That means if you have 500, 1,500 recruitment and a net gain of 500, that means normally you're having 1,000 bees a day die of natural attrition.
Right.
So there's this very dynamic process during this buildup phase. If you got 1,500 bees a day dying from attrition, there will be no growth at all. If you have
Yeah.
1,600 bees dying from natural attrition, then it's going to go downhill. So this, if a pathogen causes the bees either to initiate foraging at an earlier age or to do altruistic removal and not come back because they feel sick, then you're going to have colonies dwindling. So I was disappointed to hear that today, that a number of colonies are not building up as we would expect. Normally, when we come out of almonds, we've had good weather pretty much in the almonds, plenty of pollen in the almonds. Colonies usually come out bursting at the seams from the almonds right now. To hear them come back, say they had to look through 200 colonies to get 20 that were bursting, that's disappointing to us. Now, what you got to realize when you talk about all these losses, maybe half of the losses were expected. There were beekeepers telling me last spring it was going to be bad because it was a very good year in almond pollination. The bees came out of almonds very strong, which means a lot of beekeepers did not get on mite control early in the season, split their colonies with too many mites, and then got honey supers on them without getting the mites under control. Once they got honey supers on, most beekeepers don't do much. And then in the late season, I get reports that, hey, we took all our honey off, we're treating for mites, and we're seeing a lot of beekeepers with their honey supers still in their hives trying to get that last 10 pounds of honey. They said, those colonies will never make it through winter. We can guarantee that because they're starting mite control too late in the season.
Mm-hmm.
So there were a number of beekeepers in late summer, well, starting even midsummer, saying it's going to be big losses. By late summer, there are a lot of beekeepers telling me, Randy, it's going to be bad this year. It's going to be a lot of losses. And that would be for varroa. So I would not have been at all surprised by 50% losses this year, considering we've been running in that 30 to 40% range.
Mm-hmm.
But on top of that, we're seeing this other thing going on too, and so it exacerbates what would have already been losses, but these extra losses tip it over.
Mm-hmm. And I was listening to, I think it was Greg talking to Bob Benny, and Bob mentioned, he said he almost compared it to, and this was something that you had said, and I'm not sure when, but almost compared it to a leaky boat. And, your boat, you can have a couple small little leaks in it and you can somewhat manage it and you'll be fine. But in that whole entire scenario, you throw one more item, one more kind of kink, one more leak in that boat, and the whole ship sinks.
Yeah.
And that's almost like, where we're at now is we're hitting that tipping point where, well, did that final leak spring?
Mm-hmm.
And like my yard, I started to see a decline as far as my numbers about, 16 months ago, I was sitting around 16 colonies.
Mm-hmm.
And I lost 50% last winter. And that was actually a high loss for me. The prior 2 years, I was, I would have around upper 80s, around 90% survival. So when I took a 50% hit, I thought, wow, it was kind of shocking. Now, last year here in Northeast Ohio, our season started about 6 weeks early, where we would normally not see swarms until we're talking third or so week of May, fourth week of May. One of my colonies swarmed April 11th.
Okay.
And when I was pulling in, and it's just, last year it was abnormally warm here.
Right.
We had activity in February. And that's just so exceptionally abnormal. That's not a normal year. So our season started early, and I thought, well, that's fine, and I normally do put feed on. I put feed on them. I put pollen patties, so I like to supplement them. That way, I'll help boost them. And when we went from starting early, our April and May, we recorded twice the average precipitation. So we had so much rainfall early that it started out, and we had a lot of queen issues. Queens weren't getting mated, they were failing. So we started out early and then had queen issues. And then after May, we went dry. And Ohio recorded, I think, down kind of where central Ohio, I think it was the worst drought on record in, it was 90 or so years. Our year last year, we had so many factors that it just, this area, I've talked to a number of beekeepers. I know there's one that he had 45 colonies going into this winter. He's now sitting at 2.
Wow. So let me ask you a question. Did you do any mite washes in the spring? What did you find when you did the mite washes?
So my highest mite wash last year was 3.
Okay.
So wow. I use, and this was always been my thing, I always used Apivar early. I always put that on earlier just because it's not as harsh of a treatment like Formic or some of those other ones. It's just, hey, as the colony's building, let me get a treatment in, leave it for 6 weeks, keep them kind of under control.
Yeah.
And then after that, I use oxalic acid. So I treat several times throughout the year.
Okay, so it wasn't mites then. It wasn't. But that one thing, the reason I ask, when you have an early start in the season, that means the mites get a head start.
Yeah.
So you're gonna expect mite counts double of norm if they have an extra month in the because they double roughly once a month. So if you start a month early, your mite counts are going to be twice as high to start with, and then. But if you so you can rule out that it was mites then. So that's interesting that you saw that happening. What I would like to see is so an epidemiologist would have a map up right now and say, where are we seeing what symptoms? Mm-hmm. And then you see, and who's moving bees from where to where? Can we see this spread? Back with CCD, you could track the spread of CCD happening. It wasn't published because nobody wanted to embarrass the beekeepers, but there were certain beekeepers moving, and they just left a trail of this infection. From the Nosema ceranae or the viruses behind them. The thing is, we didn't realize that we even had Nosema ceranae until well after it was over. It invaded around 2001. It wasn't even identified to 2006. The name CCD wasn't even coined until 2007 when Hackenberg made it prominent. But it started happening around 2002 or so, 2004, 2005, that winter. That's when we came up, the industry came up short for almond pollination, and the growers tripled the price they offered for bees. So when they talk about CCD and say, oh, yeah, it happened in 2007, no, it happened the winter of 2004, 2005, and then got even worse for a few years and disappeared by 2009, 2010. So we may have, it's not unusual to have plagues every decade or two go through bee populations. This has been recorded in various parts of the world for as long as you can look back at history. And many times the plagues just are unidentified. So I'm looking right now at a couple of viruses of interest.
Okay.
Yeah, I'm not going to say anything yet, but there are disagreements. There were disagreements between some researchers during CCD about who was testing accurately for different viruses. So I'm going back for that and actually collaborating to see if some of these viruses that we have had warning signs of before, which are not on the normal screens for viruses, if one of them might be involved.
Oh, okay.
Yeah. So the thing that happens is you can see it with tracheomite when it invaded. You see it with Nosema ceranae. We see it with chalk brood. We see it with European foul brood. You get a new variant. Just like with COVID, You get a new variant, and all of a sudden, people who are immune no longer are immune. You get a wave of infections, and then people have immunity built up, or they get vaccinated, and then you don't think about that one until you get a new variant come along. You see this with chalk food. You'll hear, well, everybody in my neighborhood's got chalk food. They have it for 2 years, you just can't get rid of it, and then it disappears. And it's not because of anything what anybody did. The bees apparently build up immunity, and they can either do it genetically or transgenetically through what's called transgenerational immune inheritance. And I think this is something that's not been studied enough. That when a mammal mother starts nursing her baby, the first milk that comes out is called colostrum. In that colostrum, she primes the baby's immune system with for the target of the pathogens that her body has been exposed to. It appears to me that the honeybees do the same thing via the jelly. We know that the queen can do it to some extent. That's what the Dallin vaccine is based on. But I can't say for sure, but the evidence is fairly strong that the nurse bees can do that via the jelly and prime the immune system.
Interesting.
So this is maybe what happens with tracheomite. It was genetic. When something is killing a large proportion of your population, natural selection takes place, and there were already existing in the bee population a proportion of the bees that had genetics for their setae, their hairs around their spiracles, to harden up a little earlier than the norm. That prevented the immature or the mature tracheomites from invading the spiracles of a newly emerged bee. They can only invade the spiracle of a newly emerged bee. If those bees hardened up their hairs from the spiracles very early, the tracheomites can't enter it. So within a few years, natural selection just bred for bees that had that. Tracheomite disappeared, independent of anything that the beekeepers did. Same thing with Nosema ceranae. During CCD, on one of the chat groups, one of the beekeepers said, "Hey, is anybody breeding for resistance to CCD?" And I said, "Well, to the best of my knowledge, none of the queen producers are breeding off of dead colonies." So the answer would be yes.
That's very true.
Not because they know what is causing it, but because you don't breed off of dead colonies.
Yeah, that's very true. Yeah. We had, there was one question in here from David. It says, Randy, are you familiar with the iridescent virus?
Well, David must have been reading my mind. We are deep into looking for that, David. Yes. During CCD, I was good friends with Jerry Bromashank, and they were working with the Army, doing they had an Army lab that did advanced proteomics. And they found that they said iridescent virus was prominent in the samples of bees collapsing. In my operation, I saw something I had never seen before, and that was wax moth larvae dying in the hives. Normally, wax moth larvae thrive in my dead outside. I'd never, ever seen a sick wax moth larva. But here's the thing. Some of those wax moth larvae were bright purple. And if you are in the business of raising wax moth larvae for fish bait, your biggest fear is iridescent virus, which turns the larvae bright purple. So with my very eyes, in my operation with collapsing colonies, I saw purple iridescent wax moth larvae. So that means that my mind has been open since then to that. And I also spoke to the world expert on iridescent virus, who's from Mexico. I followed up on that, and we are reinvestigating it right now.
Wow.
Interesting. Wow.
Here, did I mute you? Must have muted you.
Randy, I have a question.
Yeah, go ahead.
I have a question for you about early spring treatments. My bees right now are looking pretty good. I had an absolute infestation in the midsummer last year. I pulled the honey supers off, and I treated pretty well through the winter last year. I did not, kind of as you mentioned, I did what a lot of people do. I stacked my supers up, did not treat in the spring, and I thought I was in good shape. And then Auburn University came down and did a study of apivar resistance or amitraz resistance on my bees, and they tested out good. My bees didn't have, like, they supposedly they were okay with the apivar resistance, and I'm not sure if you're aware of the study they're doing, but it's pretty interesting. And
I'm very much aware.
so I thought, this is awesome. So I threw apivar in there. They came out actually like 2 days before we harvested honey in my summer harvest. That's my biggest harvest, and we put the apivar strips in immediately. We condensed the colonies down as we pulled honey. We put apivar strips in, left them in there the correct amount of time, and then pulled them out. Didn't test immediately after we pulled them out, but I went through, I think it was late September, and tested, and I had some mite counts that were off the charts. Now, a lot of them were just fine, but I had a few that were like, just, I've never seen numbers that high. And so I hit them hard with Apigard. I did a full Apigard treatment. It was kind of cool that day. It was within range. And then I did all of them. Then I did an OAV treatment the next week. And then I went in 2 weeks in a row and put a half dose of Apigard in. It was warm again, and knocked the mite loads down. And then in December, I used OAV 4 treatments, with 4 grams per box. I just hit them hard. Yeah. Right now, the numbers actually look pretty good. The bees are looking really good. But my question is, I know, and I see Ian's in the comment here at Canadian Beekeeper blog. He talks, just as you just did, about the importance of having mite counts way down right now. Early season for us. I know we're a little bit delayed in the northern states, but what do you recommend? Like, for example, in my case, I've just been in the process. I've split a lot of my colonies. I'm trying to get this attitude under control as far as mite treatments right now. What would you recommend? Because here we have a pretty early honey season, like in April, late March and April, we start our honey flow, and it goes through mid-June or so, and then we're done. What kind of thing would you do now to help with those get those mite numbers down to help prevent that viral load?
Well, I don't make recommendations, but I can always say the pros and cons of various options. So, Apivar, that's used because it takes a long time, takes 8 weeks for full efficacy. That's a good treatment during the spring if you want to use a synthetic miticide and if your mites are not resistant. Sounds like some of your mites in some colonies are resistant, right? Now, you're doing the right thing, though. If you rotate another mode of action, then that strongly selects against resistance. So it's the beekeepers who do amitraz that are breeding for resistance by simply rotating. And I'm actually been modeling this. I have a model I've developed to demonstrate. I'll be publishing it to demonstrate how resistance happens. So rotating in other modes of action should breed very strongly against those resistant mites. So Apivar is one in the springtime. Another one would be formic acid. The reason is that if you have a queen issue, and that doesn't have to happen, formic acid, if you don't give them too big a flash on the first day, so if you're using like Formic Pro, if you simply put something impervious over the top of the strip, not on the bottom, but the top, that reduces that first day flash, and you don't wind up with queen loss. Even if you do, it's very easy for the bees to replace the queen in the springtime. So Formic, that's another good one. Then Oxalic doesn't do much during the springtime because there's too much brood, unless you do splits with a queen cell. So that's what we do. We split our colonies, and we actually split them down to nukes. But if you were a small scale beekeeper, you take your double deep, and you pull out the combs, and you put any combs of sealed brood into one box, and combs that do not have sealed brood in another box, and make sure the queen gets into one with no sealed brood. If you don't see the queen, shake all the bees into the box with no sealed brood, put a queen slit over it, and put the other box back on top. In half an hour, the bees will walk back up. Now you have one box that has all the sealed brood and a bunch of nurse bees, but no queen. You move that to the side and you give them a queen cell. The box that has the queen, leave her in the home stand. You don't really want to leave her very many bees because all the field force is going to fly back there. So she is in full laying condition, a bunch of empty cells with a field force, and dribble her with oxalic acid or vaporize it that day. You're going to eliminate almost all your mites. You stop swarming, and you've kept that queen. The other half has all that sealed brood, which is going to emerge in 18 days. The queen with the queen cell will not have any larvae that are 8 days old yet. At 8 days, that's when varroa can enter. So at 18 days, put on your calendar, you go back and you dribble them or vaporize them at that point. Now you've controlled mites in both halves. You've requeened one colony, and now you can. So you've kept them from swarming. You've got a fresh queen. You've controlled varroa, and you can solve the marriage issue. The marriage issue is when the spouse says, I thought you said this was going to be a hobby. You got 24 hives in the backyard, and now you're going to split them all in half. So what you do, you just put them back together. As soon as swarming season is over, you put them back together. Don't even bother looking for the queens. Let the bees choose which one of the queens they want. Now you have a super strong colony with no mice that give you a big honey crop, and you haven't increased your colony numbers.
Wow. There you go. I like it.
Okay.
I'll have to go back and watch that section again because that was some good stuff right there.
Yeah. Yeah. Wow. Yeah. Ian did, I think he's probably still in here. He did have a comment here that I want to pull up. Have a friend that is removed from modern day agriculture who has experienced losses. I mean, there's a lot of people that experience losses.
And this is why I would have an epidemiologist looking at the locations, putting them on a map and saying, hey, is this an area that was near this crop where this was sprayed? Is this an area that this or that? I was curious about who's feeding what pollen subs. A lot of are saying, hey, we're feeding pollen subs when nothing's happening.
Yeah.
We could rule that out immediately, pollen subs, as a possible factor by simply saying, hey, what if you had healthy hives, what sub were you feeding? If they were feeding this brand of sub, then you say, okay, rule out, it's not pollen subs.
Right.
Okay, if they're seeing it also in non-agriculture areas, such as mine, I can tell you it's not pesticides in my area. We got no exposure. We got 55 yards, none of which have exposure to pesticides. So it wasn't pesticides for us.
Yeah. I know there's, and I know there's so many people involved in doing their own kind of research. They've got a lot of different surveys going on out there. There's the one individual that I know, Rob Pollock, that is with Lorob. He has a survey going, and he just threw it out there. And I would have to look at the exact data points of what information he collected. But I think like in the first day, he had a couple hundred individuals. By like the end of day 2, there were like 400 people that had submitted data. But like right there and now, I think he posted a link towards some of his initial data, but like right there, it's saying high losses from Florida, Michigan, and Maryland.
Right. So that's an epidemiologist. Now, I'm not an epidemiologist, but I do understand epidemiology somewhat.
Yeah.
The first thing I would want, you think of this, if it is a new virus, which it has a very high probability of being a new virus or a variant of a virus, it would have had to evolve somewhere, and then it would have had to spread. Now, if we didn't have any movement of bees, you could track the spread very easily.
Right.
But since we do move it, how's it going to move? It's going to move by beekeepers who are moving from Florida to apples up in Maryland or something like that, or somebody selling packages or nukes. So let's just say that the supplier for one of the big package suppliers happens to have their operation. Well, now you're going to immediately have it spread to anybody who bought those packages. And it's not that you can blame them for anything wrong. Obviously, bees that are, you don't shake bees from dying colonies for packages. So it's not that they knew something was happening. But if this new virus was out there evolving and then it gets spread, now you look back to, okay, so how are these different locations related? Did they buy nukes or packages from the same people? Was there migratory beekeepers moving from Florida up to this area? That would very clearly, if we can see transmission routes, then that means it's very likely a pathogen.
That's one thing that I think is interesting, and probably in our country, I would imagine more than anywhere else. the migratory beekeepers are it's just a huge part of it. Bees going all over the country from all different parts of the country. And so I don't know. I think that there may be a lot to be said for what are they picking up something when they're in California with the almonds? Are they picking up something wherever they're at? Personally, my bees, I sent some out to almonds for a few years there, and I haven't done it lately. But when it comes to the question of sprays and agriculture, almost all my bees, my healthiest bees, are right in the middle of big cotton fields and peanut fields and all kinds of crops all around, and my bees typically do well. And so
I hear that a lot.
I mean, it's it's as long as they have a good water source and the type of trees that produce the pollen and nectar we need around here, they thrive. I actually have a harder time with my bees that are in locations where there's not agriculture. It's kind of crazy. If there's just woods all around, they seem to struggle a little bit more. Certain times of the year during the honey flow or during the nectar flow, they tend to do really well, because those trees are located. But other times of the year, it seems like during the winter time or during the certain times of the year, there's just not, when there's no cotton blooming or no.
Oh yeah.
Our fields have what we call yellow mustard around here, just acres and acres of it. That I guess it's a cover crop. I don't know if it grows wild. I think it kind of just grows wild in the off season, and that's winter time. So that is a wonderful source for pollen, maybe even nectar, and it's everywhere. And so, I know that there may be something. It may just be a cumulative effect of all the different things together. But I just wonder about moving bees around, and the Florida bees going to California for almonds, or the Georgia bees or whatever going out there, just mixing and flying around with all the other bees. And are they picking stuff up? Are they picking viruses up? Are they picking different things up in that? It's just a huge question.
Yeah, what? I've pollinated almonds going on 50 years, maybe more than 50 years. And we just don't see that it being a problem. So understand that anywhere you put bees, the moment a bee lands on a flower, it leaves virions. The next bee that lands on that flower picks up virions. Transmission is almost instantaneous. So it's anybody who's moving from Florida up the East Coast doing pollination, they're going to be spreading them. So targeting California, obviously that takes stuff it used to take a while for stuff to move from the East Coast to the West Coast. Like we watched trachea might slowly move across the country. Nowadays, anything's going to be distributed, has an option of being, or possibility of being distributed within a year.
Absolutely.
So, but we can, again, use that epidemiology to rule out that it's a pesticide. Okay, I don't see any sign that it's a pesticide.
Randy, a question I'd have for you. You mentioned the epidemiologist. How does a standard, like a sideliner like me or a hobbyist like Brian, how do you get your bees in front of an epidemiologist? I know I have my local bee inspector actually takes studies and sends them up to Maryland pretty much every year. I know they do. They have a big lab up there. Is that what they're doing up there? Are they looking at the different viruses? I think they are looking at the different viruses and things.
So like Brian, a sample of his bees would be of great interest. The problem is RNA viruses. There's RNA viruses and DNA viruses. DNA is very stable. RNA is not very stable. So DNA, you can take dead bees and pick it up easily. RNA viruses, they generally want the sample taken and put on dry ice and shipped immediately. So I did deliver bees on dry ice this last week. So in his collapsed colony, that would be of interest because that's an atypical collapse. That didn't sound like starvation. You had your mites under control. Mm-hmm. Another thing that we're seeing is Declan Schroeder, the virologist, at a recent conference said there are some recombinant strains of deformed wing virus that are extremely virulent. We do not see deformed wing virus in this right now, but there is something happening out there. I've seen it in our operation, and a number of other beekeepers have told me they're seeing colonies with bees with deformed wings, and the colony starts to dwindle. And I go out there and go, wow, let me go back and get some dry ice and come back tomorrow. And by tomorrow, they're all gone. It's just like I've seen it again and again. The colony crashes so damn fast with deformed wings, but no mites. So apparently there are some recombinant forms of deformed wing virus that are extremely virulent. It doesn't sound like that's what's happening now. Of course, I haven't seen I can only speak for looking at my own bees. But
Yeah.
the way a hobby beekeeper can help, if you have a colony in the middle of that collapse where there's still some bees alive, you can get any researcher to let you mail them a sample on dry ice or willing to come out. That is good data. The problem with CCD is many of the samples were taken forensically after the fact. The researchers showed up and you see a bunch of dead outs. You can't learn much from a dead out. Our
Right.
advantage with CCD, I had Dr. Eric Musson as a monitor of our trial, and where we created it by inoculating the bees with Israeli cuprous strain that we isolated from another operation that was in collapse. I cultured the virus in bee pupae in an incubator
Wow.
and then had that virus purified. And then I got the pure inoculum sent back to me. And then did cage trials to find out how much you had to feed the bees because normally it's hard to infect them with the virus by feeding. It was dynamite. I mean, that tiny amount of that inoculum, it would kill the bees in the cage. So we inoculated all the bees in this trial and then used RNAi that suppressed Israeli acute paralysis virus, and we could see a big difference. So it was the combination of the virus and Nosema. Anyway, at final grading, Eric Musson came out from UC Davis, and there were 72 colonies in the trial. At the end of it, he sat down with me on a couple of beehives. He said, Randy, I've heard about CCD. I've followed it all. I've signed off on operations from beekeepers who needed a sign off so they collected compensation for CCD. He says, I've read all the papers. He said, there was nothing like what happened today. I got to see it at every stage of progression right before my eyes.
Hmm.
And that was fascinating. So I went home, thought about it, and I published a model of how CCD occurs. And it was with 4 positive feedback loops. And the researchers at USDA actually adopted that model of mine to explain. So it is kind of like that leaky boat type of thing, but you wind up with this a combination of Zima viruses or whatever things, and by affecting the adult bees so that they don't have as long a lifespan, the whole colony then dwindles down.
Wow.
So much to think about.
I know.
Oh yeah,
I know. And I think like when you mentioned Nosema, it's been probably 3 years now. I've used Hive Alive supplements in my operation.
uh-huh.
So, when I feed them syrup, they have Hive Alive in it. Whenever I give them pollen patty, it's the Hive Alive, so it has the Hive Alive dosing in it. And Hive Alive, they say, is supposed to help prevent nosema. So, I just kind of scratched my head this year. It's so puzzling at the
So
colonies.
so here's how you stop your puzzling. You take half your hives and you feed them Hive Alive, and the other half you don't. You get a microscope and you see which ones have nosema.
Right.
Okay.
Yeah.
So there have been trials run, and
And
the manufacturer may interpret those trials differently than other trials.
yeah, and I just, within the last month, bought a microscope.
Right. So like Bruce might say, I give my bees Jim Beam whiskey every month, and I don't know if I have nosema problems, but I don't know a microscope. Well, I believe Bruce as well as I believe you.
Yeah. Yeah.
But how do you know if it really helps or not if you don't have a control group?
You have to have a control group, and that's
So-
that's the number one problem that beekeepers don't do. They try something on all their hives, and they don't have a control group. So, example,
Yeah.
I often say this when I have an audience. I said, well, so people ask about black queen cell virus, which affects us when we're bringing queens, kills the queen cells. I said, well, we had it really bad a few years ago, and I asked the other experts. They said, well, it's treated with fumagillin for nosema and terramycin. I said, well, terramycin is an antibiotic, not an antiviral. They go, yeah, we know that, but that's the common recommendation.
Yeah.
And so I said, okay. So I mixed up fumagillin and terramycin, and I fed it to cell starters. And sure enough, a week later, there was no more black queen cell virus. And I asked the audience, what did I learn?
Nothing.
Well, no. What question do you need to ask me? What happened in the control group? No audience ever asked that question. Any scientist, that'll be the first question you ask. This is the problem with beekeepers. No audience ever asked the obvious question. It disappeared in the control group too.
Yeah.
So that means I didn't learn anything.
Right.
But if you don't run the control group, you're going to say, oh, that cured it. That's the number one problem that beekeepers have. They do not run control groups.
You don't know if it would have cleared up on its own had you not treated. I guess that's the.
We do know question. It did clear up on its own without treatment.
Oh, okay.
Yeah.
Go
ahead, Bruce.
I have a local commercial beekeeper. He's a friend of mine, kind of a mentor of mine. And in describing his loss, I personally, my bees look very similar to where they have in the past, maybe better than they ever have, or very similar. But like I say, I really got on the mites. Now, I did have a problem last summer, but I jumped on it and I hit them hard. I mean, I just hammered the bees, and I lost some, but maybe those are the ones I needed to lose, if that makes sense.
So here's how you're going to help. You go over to Brian's, you find the colonies collapsing, take bees from that and add that to half your hives and mark those. And see if it's a transmissible disease. That would be very helpful.
Yeah, it would. But Tyler says, I asked him, I sent out the question back a few, I guess it was on Valentine's Day, a couple of weeks ago. I said, have you experienced the loss that people are talking about? And he's here local. He's real close to me here. And he says, I feel like he says 50 to 90% losses, no. Yearly 30 to 50% losses, yes. I felt like my mite treatments were better and more effective this year, but I still think I lost about the same as 2023. I still see losses that blow my mind. All signs of perfectly healthy colonies, and come back a couple of weeks later, and they're a complete loss or shrunk to a few frames with a queen still. I really don't know what my exact percent loss. Maybe I should figure it up, but I have more bees now than I've ever had coming out of winter. So he's having that kind of similar thing to where they just kind of dwindle down for and he's really, he really works. He's a commercial guy. He's a young guy, and it's his livelihood. And so he is really working hard to try to take care of his bees. He feeds them, he treats them, he sends bees out to almonds and everything. He does all the things. And his bees look typically really good, but just the thing that has him scratching his head is, why are they healthy one day and 2 weeks later they're dwindling, or they just some are complete loss, or they're
that's
just
us too. So that is our question also.
Okay.
So that's why I'm suspecting a pathogen.
Yeah, makes sense.
Yeah, we had a question here from Mike, and Randy, you may know him. I'm not sure. He does.
Yeah. Hi, Mike.
Yeah. How's your titration testing going?
It's going great. I'll be publishing it soon. I've got 5 years of work, intense work on oxalic acid that I presented to groups, but I've never published it yet. So I'm going to hopefully next month start publishing it. It'll probably be 12 series of articles of going through everything about oxalic acid. So Mike was generous, I'll tell you right now, with his sent me some of his pellets. Well, if you're using vaporizers, those 1 gram pellets are really, really handy. Well worth the cost. So, so I was quantifying the amount of oxalic acid that actually comes out of a vaporizer, because you have thermal degradation that happens when you put it in there. And tested 4 different kinds of vaporizers. And if somebody has another kind of vaporizer they would like me to test it, they want to just send it to me, and I'll test it and send it back to them. I can't buy every vaporizer on the market, but I'm happy to test them. So we do a titration. We go ahead and we take the vaporizer, follow the manufacturer's instructions. We can put in we're putting 4 grams of oxalic acid into it, and then we have a capture apparatus that captures all the vapor that comes out and recondenses it, a chilled condenser. And then we can rinse all the acid out of that and then titrate it and see what proportion of that 4 grams actually comes out of a vaporizer. And something that I had learned 4 or 5 years ago was I did an experiment 4 or 5 years ago, wondering about how much comes out. And I took vaporizers, and you can put a balloon over the output of a vaporizer. And you've seen that giant cloud that comes out. Well, if that balloon is chilled, I used a Mylar balloon initially, it immediately condenses when it goes in the balloon. The balloon does not inflate at all. Then you get to 210 degrees centigrade, and suddenly the balloon inflates a little bit, and it stops inflating at 220 degrees centigrade. The total inflation is absolutely minimal, but a little bit of inflation remains. And that means that any oxalic acid that comes out will immediately recondense into oxalic acid. Any water vapor that comes out will immediately recondense into water, so the balloon will not inflate. So that means there's something else that is coming out. And so I suspected either formic acid, carbon dioxide. Found out it's not either formic acid or carbon dioxide to any extent. It's carbon monoxide. And you can test it very easily because you touch a match flame to it and boom, it burns with a beautiful blue flame. So the oxalic acid vapor doesn't burn at all. Formic doesn't burn at all. Carbon dioxide doesn't burn at all. And it only burns between 210 and 220 degrees centigrade. And then the flame goes out. So very interesting doing these titrations. They are these vaporizations into this condensation tube. So then we go ahead and titrate it, and we find out that you only get 40 to 50% of that oxalic acid that goes in actually comes out. That doesn't mean that you have to increase the dose. The dose is already determined experimentally with how much goes, that they do experimentally. But and we tried changing the temperature on them. That doesn't help. That's what you get when you put it into a vaporizer. You typically only get vapor for about 60 seconds coming out. It's very, very quick. We took a series of quart bottles. Hey, Greg. And,
hey, Randy.
Put them all into a freezer so they're ice cold, and then line them up, start a vaporizer, and then put a jar for 10 seconds over the mouth of the vaporizer, and then immediately put the next one on, the next one on, the next one on, and collect everything comes out. And it's really, really obvious at exactly what temperature and for how long you actually get oxalic acid coming out. Because the jar turns white inside with oxalic acid crystals. Below that temperature, there's no white. And above that, there's no white. And it's exactly at the same temperature you get the thermal decomposition. So I'll be publishing. I got lots of photographs. I'll be publishing it all.
That's very, very interesting. Yeah. There was a question here, and when and this is going back to the colony issues, the colony collapse, when we talk about viruses, everybody always just initially, whenever a virus is mentioned, everybody always just thinks varroa.
No.
But what about this right here? So, and Filament Corner at Spanky, he asks, does Randy possibly have an idea on how many viruses small hive beetles can spread?
Okay, one thing you got to realize is that all these this is recent research by Donna Cox Foster. She studied in I think it's Colorado, in Utah, and she worked with beekeeper Darren Cox, had him put hives out into the landscape where there are native bees, bumblebees, other native bees, and she tracked all the viruses and the buildup of the native bees and the honeybees. She found out that there's different variants of each virus in each different species of bees that are evolved to be with those species. So the thing about people saying, oh, honeybees are spreading viruses to the other species, if each species has its own variants of a virus, and we have maybe 400, there's 3,500 species of bees in North America. In any area, it may have 400 species of bees, of which one is a honeybee. Which direction are you going to get viruses spread? You're going to get them spread to the honeybees from the other bees, not from the honeybees out to the others, because they're all different variants. So the honeybee variants of deformed wing virus are unable to reproduce well in the other bees, but the other bees will have their own variant of deformed wing virus, or their own variant of sac brood virus, or of Lake Sinai virus. So that was one years ago after CCD. Michelle Flanagan asked me and some other beekeepers prior to going to almonds, pick out a half dozen of your colonies that are really strong and a half dozen that are not looking good, and send me samples. And she did. She found out, it's Lake Sinai virus. It wasn't deforming virus. It wasn't varroa. It was Lake Sinai virus, something that was not even on anybody's radar. And that recent paper just came out of Canada. That's one of the major viruses. That and black queen cell virus are out there, and sac brood virus. And sac brood virus, that's what Leslie Bailey back in the 90s in Europe said that's the number one virus carried by honeybees, the sac brood virus in the adult bees. You may not ever see any symptoms, but it's there all the time. So none of those are necessarily associated with varroa in any way. So it's like a kindergarten classroom. There was a very good paper published by Michelle was involved, Joe DeRissi, I can't remember who was published by years ago, where they tracked Brett 80's hives over time, and they showed the constant change of viruses that came and went in each colony, just like a kindergarten classroom, where it spread, they purge themselves of it, another one comes in. It's very dynamic all the time in beehives. So it's possible that a small hive beetle might carry a virus, but the transmission via flowers is probably much greater than you ever get from small hive beetle.
Interesting. Wow. Greg, how are you?
Good, guys. Sorry I'm late. Randy, nice to see you again. And
yeah,
it was great seeing you at the Bee Expo and talking with you about the Oliver wash cups and the wax dip results and things like that. And so I've been listening in on the drive back. I've got to spend the last couple of days working with a fantastic commercial beekeeper down in South Carolina. Runs about 1,000 colonies, and we spent the last couple of days going through a bunch of colonies and adding boxes and adding feed, and his colonies are looking fantastic. And he's
wonderful.
Spent a lot of time.
Lucky guy.
Well, and it's great. It was such a blessing to be down there, being in the thick of so many healthy bees. It's so interesting as we're all trying to wrap our heads around what's going on and have conversations with beekeepers, and we're not looking for the answer right now. We're looking for feedback. What were you seeing? What were the outliers? What was different this year than other years?
And a big question: did he have any exposure to other operations? Did he bring in any packages? Did he move his bees elsewhere for pollination, or was he relatively isolated?
Nope, he's a migratory beekeeper and sends his bees to the almonds every year. So he would be the typical person that everyone is pointing a finger at and saying, you're part of the problem, you're part of the cesspool. And that's not, and he's going up and down the coast all the way up to California and back, and that's not happening there.
Yeah.
What was interesting is, so his bees are looking great. I just came back from Alabama, but maybe a couple weeks ago from Bruce's place. His bees are looking great. But one of the things as we're talking with customers and hearing all the feedback is a lot of these reports are occurring in pockets where there was absolutely extreme weather events, one way or the other, either extreme flooding or extreme drought, all the reasons why, and potential stressors. And as I'm thinking through the year, some of the really wild things that I haven't seen in 10 years is this extreme drought. Like Brian, I was listening in, we're a D4 drought for 14 weeks, worst drought Ohio's ever seen. Plants weren't producing. Goldenrod, which we lean on heavily, didn't have enough moisture to create nectar, but it went into survival mode on pollen. But here's one of the interesting things. And I didn't hardly see anything, not just the honeybees, but no pollinators on the goldenrod, on all three varieties this entire fall. And to me, that was a big red flag that I'm thinking now is, if they're not even touching the goldenrod or the asters, what is going on here?
Or they weren't there.
or they weren't there. And here, and so as you guys are talking, I'm thinking of all these things that I wanted to bring up, Randy. But a lot of what you're seeing, and a lot of folks are seeing, is what we had beautiful colonies early in the year. We were monitoring heavily. The only colonies that ever got out of control, believe it or not, were VSH colonies, and the F2 and F3 hybrids. They went backwards in time.
Okay.
We removed those from the isolated quarantine yard. That's history. But all of our regular running line stock was doing beautiful. They were keeping mites at a 2% threshold or less on their own, but we still went in with formic. We still went in with OA, got everybody cleaned up. They were building up tons and tons of stores. We got so busy here with the wax dipping stuff that we didn't get around to pulling all the supers. And so they were fat and heavy, low mite counts. And I'm going through looking at these dead outs, and something doesn't add up. And I'm trying to figure out what in the world is going on. But we've got, I have found the queen in nearly every single dead out, right? And she's surrounded by barely a baseball size clusters. Yet they were in double deeps with 1 or 2 supers. There's some of them 70 to 120 pounds of honey still on the colonies, but yet there's only this many bees, and the bottom boards are barren.
So, like, what you can do, and you can do this with those bees. That handful of bees, put those under a scope. Look at those bee by bee, one bee at a time, and they're going to be either solid nosema. You do a gut squash on them. And each individual bee, it's either going to be more cells they're going to even imagine counting, or more spores, or none. It's black or white.
Okay.
And I did that with ours. I'm not seeing it, but that's the symptom of nosema disappearance. So if you could get any of those silver dollar patches, those would tell us a lot. And let me know the results. You put them in an envelope, dry, and send them to me, and I'd be happy to squash them and tell you. So that'd be very valuable samples just to be able to rule out whether or not it is nosema, because that was
it's uniform is the thing. It wasn't just like we have 1 yard of 100 that we lost 98 of the colonies in, and only 2 are surviving. And all 98 that I went through, I nearly found the queen in every single one. It was extremely with a year of inconsistencies. This is extremely consistent. Tons of honey. But no, where's all the bees?
That was CCD also. That's the same symptom that we saw on nosema and fibrous collapse. And that's why I'm not confused, but surprised that I'm not seeing nosema in my operation. So, man, if you could send me individual, put them in a Ziploc bag, just dry.
Sure.
Give me a few samples. I'll let you know whether nosema is involved in your operation.
Okay, yeah, well, I'll do it.
I have one of my remaining colonies. I have one that I know it will probably be dead within a week, because it's that exact same scenario. When I went out today, it was the cluster size is like my fist.
Oh yeah.
And it's just like,
you have a microscope now, right?
Yeah.
Use it.
I know I need to because
it's really easy.
I went and I got like an entomology dissection kit. So I have all of the tools. I need to dig into it.
You don't need to dissect. You just take a few live bees. You just put a blade or a spoon on their abdomen and just roll it. Squeeze their gut out. Put a cover slip over that and take a look under the. Might have to add a tiny droplet of water under the scope. And you're either going to see solid spores or none.
Okay.
It's really quick, really easy.
I'll do that. I'll have to do that this week.
Randy, so let's say we get, so you're saying you're not finding nosema, but you're finding these tiny clusters. So that's leading you.
No, we're not finding tiny clusters. We're finding dwindling down, but they're not going down to tiny clusters. They're going down to like 2 frames of bees. So, but during CCD, that was I took a photo, pulled up a photo today. I'm writing an article that shows that exact symptom. And what you can see is the on the frame here, you see the ring of bee bread and the last cat brood, and then this little in the middle, this little tiny cluster where you can see how big they were a week before, and now they've shrunk down.
Yeah.
Yeah, it has been extremely cold up there too. So back to Greg's point, I don't know if that would have anything to do with it or not. If they are naturally brooding way back because of the cold, and then
Cold's a big stressor.
there's some other external force that's making these bees brood.
Yep.
Just they aren't strong enough to withstand the cold.
Yep.
Yeah.
Perfect storm. I think Brian touched on it, but Randy, when I interviewed Bob and at a conference not too long ago, he brought up the Randy Oliver leaky boat syndrome and kind of laid it all out there. And I think, that's a lot of, we've seen so many different, if there was drought and poor nutrition and the list goes on and on and on.
Yep.
But it seems as though these things were an onset that really we didn't see any evidence of until getting into September, October, the first part of the entire year. So if it's like, let's say it was pathogens or it was something brought up with migratory beekeepers or packaged bees, like why would that only show up at the very end of the year, 3 or 4 brood cycles later?
Let me do an experiment. Can you help me with the experiment? I want
Sure.
you to go out and start sleeping outside with no clothes on and stop eating. See if you happen to come down with any diseases.
Yeah.
That's
True.
just put a little bit of stress on you. Go ahead and film it. Because everybody would love to watch this and see how long you survive. Okay.
Yeah.
Okay. Not enough that you're going to starve to death or that you're going to freeze to death, but just stress your body.
Yeah.
Immune
See what
response to stress. Yep.
Well,
Not response. It's immune failure.
Immune failure. Yeah. Failure.
Immune failure because the body is stressed. Okay. And
I work
so-
in a. I'm sorry. Go ahead. No, I work in a hospital setting, and of course we see patients, and that's what many people are dealing with during the more difficult times of the year weather-wise. During the cold, there's more illness. It seems like more sickness with things such as the flu. RSV is really a big thing this year. Back even when it seemed like maybe even when COVID was at its prime, stressed people, stressed systems for the most part. If you're stressed, whether it be mentally stressed, physically stressed, stressed in whatever way, it does tend to weaken your body, your immune system, and you are more susceptible to diseases. It seems like, cancer patients that are on chemo and radiation are just at really high risk many times for more disease because their body it's just not as optimal strength. And so that makes total sense that the bees, if they're stressed as well, whether it's from whatever the cause, they are more susceptible.
Yeah. So like Greg, you were at the expo with me. We have 3,000 people there.
Right.
3,000 people. You have probably are exposed to every virus known to man.
You're right.
We all were. Every time you breathe in, you're inhaling all these virions. It's not that you don't, and the same with every beehive. These pathogens are always present. You're just waiting for the immune system to go down. And
I see.
Yes.
And so it's the stressor is the catalyst that jump starts this sequence of decline. And that makes a lot of sense, and that would be in line with what we've seen. All of a sudden, folks in Ohio are understanding why Ian Stepler puts his colonies in sheds or windbreaks or all these things, because all of a sudden, over from January 19th through 25, here in our local area, we were actual temps, nighttime lows -5 to -18 were actual lows, with wind chills -25, 40-degree winds for 2 days straight. All of a sudden, Ohio beekeepers were northern Canadian beekeepers. Not only are we not prepared, but the bees weren't prepared to go a 60-degree temperature change in 24 hours. That's a major stress point.
Absolutely.
And it didn't matter if the colonies we have insulated hives, we have regular wood hives, or on 4-ways. It was a smorgasbord, and it didn't matter. It was that weather was no respecter to the type of hive the colony was in. It crushed them, and so it was all these little things. Here's the thing where this gets wild is what if we are just seeing that stressors? I think it's my opinion that bees are a great litmus test, soil health and ecological health. What are the chances that we're already in a compromised scenario with the bees, and across the country, nationwide, all these different stressors are acting like catalysts, exposing the Achilles heel of the honeybee right now. What's the chances that you're going to find 14 different things? It's not just one thing plaguing all the bees all at once.
Right. No, I completely agree. Again, that leaky boat. Understand also why you don't die when you inhale all these virions, or when you eat them. Every time you pick up any kind of food that's been exposed to anybody's breath, you're getting them. When you get a virion uh, or any pathogen into your body, either by your mouth or by your lungs, it's going to hit the tissue, the intestinal villi or the intestinal epithelium, or the epithelium within your lungs. It has to infect one of those cells. So it has to somehow get through that cell membrane into that cell in order to replicate. What your body does, those individual cells recognize that this thing should not be here. So that cell does what's called apoptosis. It immediately sacrifices itself. It bursts open before that pathogen can reproduce. So like a few days ago, I had breakfast and then I couldn't eat anymore that day. I just didn't feel good. And then that night I just barfed everything up. And then after an hour, I voided everything out the other end. It was apparently norovirus. Okay. Why? What's happening? What happens is your body, you get something into your intestine and it goes, wait, something's not right. It sacrifices every cell in your stomach lining and your intestinal lining. The ones that sacrifice in your stomach lining come out the mouth end. The ones that sacrifice lower down the intestine, they come out the other end. Your body purges your entire lining of your stomach and your intestine. That's why you get diarrhea, why you do the vomiting. It's your body purging all those cells. That's natural. That happens all the time for you. Normally, you don't get it that hard that you'll get diarrhea, but you are constantly purging all these things that come in. Nosema and the viruses, the first thing that they code for genetically is to suppress apoptosis. So if they suppress apoptosis for themselves, that means if there's another pathogen coming at the same time, it can take advantage of the other one suppressing apoptosis. So these pathogens can be inadvertently helping each other or suppressing each other. It's a very complicated thing going on all the time with the bees. And again, every time they visit a flower, they're exposed to every insect virus that's out there, every bee virus that's out there.
Wow.
Randy, one of the things that, and I got a chance to talk to, I asked Corey about it. I've asked Bob about it. I've asked Marla Spivak about it. And I've been raising the question for probably 3 years, and I'm sure you've been raising the question for probably at least 30 years. But it seems as though there is something a little different occurring over the last 3 to 5 years, that we're so focused on counting for mites and using that as the metric to remove the viral equation. But we're finding that there's colonies with maybe they're washing zeros, but they are sick as a dog, and we're looking at colonies with it looks like red sprinkles in the bottom of a cup, and they are lusterful and beautiful and clean, and they're doing all their job. Is it possible that we can't correlate mites to the potential for virus transmission, that there can be indeed occurring extreme viruses occurring in very low mite populations, and that is maybe what we're seeing, that was started maybe a couple years ago, and we're starting to see more of that evidence now?
Oh, exactly. I'm talking about that earlier with we're seeing the deformed wings. Let me answer that question quickly about microscopy. 400x, 400x. Yeah. So if you have a 10x ocular and then 40x on the objective, that's 400x. But yeah, these virus issues have been going on forever. Deforming virus was around with many variants prior to varroa. It was considered a benign virus, and it still is a benign virus in places where you don't have varroa. Varroa is just a vector for it. So having a vector changed everything. So some of the viruses use varroa as a vector, but they didn't need varroa. They were already there before varroa.
Interesting. Well, that's kind of doom and gloomish. It's not just as easy as removing the mites from the equation, then, is it?
No, that helps a whole lot, getting the mites out, because mites are not only vectors, but they also suppress the immune response of the bee.
Okay, true, right.
Sucking out the hemolymph. And something that was interesting, I never heard anybody else talking about this. There was a study years ago that showed when there's an injury to the fat bodies in an insect's body, the other fat body cells move towards that injury, which is interesting because
Wow.
that means that when varroa is injecting in the digestive enzymes to digest the fat bodies, that the other cells are going to move to that feeding wound.
Doesn't that just accelerate the?
Absolutely.
Wow.
Interesting.
Yeah.
Wow, isn't that wild?
Yeah. So varroa is a stressor and a vector. But
Randy.
anyway, it helps a lot. I just took a bunch of photos today. My sons went out to bring back our breeder queens from almonds. So these are bees that have had 5 mite washes throughout the season and have to perform very well in almonds. So they had to coat almonds strong and come out much stronger with good honey. They finally made breeder grade, and we have all these aluminum tags, the felt pen with all the mite counts and the dates on them. So I went out and took a bunch of photos today just to show. Now, I'm not saying these bees are immune to whatever's going on, but these were all strong colonies coming back from almonds. So again, we breed from bees that take care of themselves rather than bees that we have to baby.
Yeah. Randy, what in your, what does you've been working on a path forward for 30, 40 years, but right now, and the lens that we're looking through right now, what does the path forward look like for the next 1 year and 3 and 5 year? What's the path forward look like right now, do you think?
Well, interesting. I'm writing an article on that today. We've been on, our industry has been on 4 joyrides for around 20 years or so. And all 4 of those joyrides are coming to an end. Maybe not sure about the third one. That's going to depend upon a guy named Elon Musk.
Okay.
Okay. So I don't know if I want to elaborate on this because I'm going to be right. I guess I can tell you. Yeah, I guess I can tell you. So one, we've been on the joyride of Amtraz working. That is ending. Okay. That's going to make it harder for many beekeepers to deal with varroa. We've been on a joyride with the Save the Bees, and that just made the hobby market expand tremendously, which created a huge market for packaged bees and nukes and stuff, and for the manufacturers. The manufacturers have loved Save the Bees. Okay, that's pretty much getting over now. We've been on a joyride on almond pollination. Back in winter 2004, 2005, when we our first big collapse, the price for almond pollination tripled, and it went up from there. So we've been at this very high rate. Almond plantings now have been negative for the last 2 years. They're going downhill, and there are new cultivars of almonds coming out that do not need as many bees, or any bees at all, maybe. That joyride is ending. And if Elon pulls out the payments from ELAB, that joyride is going to be over. Okay, that's 4 major changes to our industry that are happening as we speak.
That would create some massive changes. I mean, completely huge pivots in the industry. I mean, not just commercially, but that trickles all the way down to hobbyists.
Yeah, what it's going to do is going to change. It's going to change things. It's going to take a lot of the marginal players that have been struggling out of beekeeping. A lot of old guys were ready to hang up their hat anyway. When that GeoCure offer came out, for $700 a hive and a new pickup truck and all the other magic, a lot of guys were really happy to take that. So this year, some of these guys experienced massive, massive losses, and it's going to be tough for them to recover. And if ELAB gets yanked, they're not going to be able to recover from that. So it's something to watch. The point I make, though, is whenever an industry, any industry, undergoes major turmoil, that creates opportunity for those who adapt and survive.
Yeah.
So you choose which side you want to be on.
To that point, Randy, got a little bit of a shift in focus here. As far as you mentioned, those that are, I guess, tough, that are willing to adapt and will survive and possibly even have, like you say, more opportunity. one thing that, you mentioned ELAP, you mentioned different things, almond pollination, along with this entire thing that's going on, if they're able to kind of shut down the import of honey from other countries, and if we have beekeepers numbers are dropping, that should increase the price and the ability to make a profit or make money on honey sales. Theoretically,
you've already done the math, huh?
Yeah, well, it just makes sense because there's so much that comes in. Like I saw a post on social media today of a guy that had some honey, and it was a local company, like a United States company, but on the back it said a product of India, and so if that can shut down a little bit, if we can just actually sell, if we can increase, so that'll allow beekeepers, like you say, to pivot. If they've been heavily invested in almond pollination, the prices of honey goes back up, then maybe we can be able to start producing more honey in our own country. But it's going to have to come from above. They're going to have to stop the imported honey, especially the stuff that's not real, from coming into our country. I'm not sure how they do that, but maybe tariffs. I don't know. Maybe tariffs. I don't know what it's going to take, but
testing. If the American honey producers is very much into this, we're learning new methods of testing for honey for purity. But it's a matter of getting the political capital to crack down on, so that these imported honeys actually do get tested. We don't have a standard of identity. Until we get a standard of identity that's enforceable, the rice syrup is always going to be cheaper than honey.
I agree, and it's even if it's just the real stuff. Let's just get rid of the stuff that's not even 100% real, and that would eliminate a lot of the market of stuff coming in. So that's where my main emphasis has been. I've done a little bit of everything, but I've done a little pollination stuff, but I just really enjoy the honey aspect of it. And I think as a sideliner, I mean, I'm a decent sized sideliner. I make a decent amount of money just doing the sideline thing. But as a local beekeeper selling local honey, I make enough and I can sell enough at a high enough price to make a profit.
Absolutely.
But when you go to that commercial range where you're having to sell it by the drum, by the truckload, those guys are really not making, it's really difficult to make a significant amount of money doing that. And so, that's where I'm at. It would just be really nice if we could figure out a way to get that figured out. I know that the beekeepers have been on that bandwagon for a long time. It's just a matter, like you say, of the political capital to be able to get the fortitude and the ability to shut it down from up above, because beekeepers can't be on the docks testing the honey. It's got to be somebody. That's where a little bit of regulation needs to come in with that stuff coming into our country.
Yeah. Also, the other thing is with the almond industry, there's 1 million acres of almonds. That takes 2 million beehives. If you didn't have the demand for almonds, you wouldn't have the stocking rate of bees exceeding the carrying capacity of the land in many areas. So the big problem now is that the number of hives in the U.S. is determined by the demand, largely by the demand for almond pollination. Mm-hmm. So many beekeepers, the number one problem is you have too many hives per acre out there. So where you used to get 200-pound crops, you're getting 50-pound crops nowadays. Okay, part of it's climate change, part of it is planting of crops, but part of it is simply too damn many hives out there. So that will actually make it better for beekeepers if we simply drop numbers overall, because it's an artificial level because it's maintained for almond pollination rather than for honey crops.
Well, the other issue too, you mentioned the environmental issues. I'm not sure about the climate change, how that works, but I do know a lot of acreage. You mentioned the almonds alone, a lot of acreage of wooded areas and natural habitats are being destroyed, and different housing projects and businesses. A lot of natural habitat is being, and pasture land even, has been moved towards that as opposed to leaving that natural flora in the area. And so that obviously hurts the bees in the area as well. When you start destroying the natural wooded areas and the natural plants that are in that area, urban sprawl can be difficult. And so that's another thing to consider.
Yeah, relative to agriculture, that's actually acreage wise relatively small. Agriculture has changed because of more effective weed control. So when I fly over the U.S. now, I look down, you look at farmland, there used to be green strips of vegetation on the fence lines between every field. Those were all food sources for native pollinators, for honeybees, for wildlife. Those no longer exist. It's a whole different landscape. Look it down. You got either monoculture crop and dirt, and that's it. There's no more green fence rows. That's a major change.
Yeah.
Yeah, I got you. Man, we're already an hour and 32 minutes in. It just flew by. It's crazy.
Yeah.
It's
Okay.
a lot of food for thought.
There's a lot of
Yeah.
food for thought. Okay, I better go get some dinner. And unless you have the last question. Questions?
Yeah, do we have any questions? Quick questions, Brian? That you should bring up.
Let's see.
Brian, while you're looking for that, Randy, thanks again for joining us tonight. Thanks for visiting with us at the Bee Expo, and of course everything that you're doing and have done for decades for the community and the industry. We thank you so much for what you and your family has done. We really appreciate you. I'll send you some dead bees, and you can tell me what you see in those too.
Oh yeah.
Yeah. This now.
Same with you, Brian.
I will.
Either do it yourself or send them to me.
Yeah, yeah, I will.
Okay.
Now, this question, this could be you could probably have a 30-second answer with this, and then we could probably talk an hour on this, but here we go. So this is from Steve, and Steve, great guy. Randy, is there any reason to suspect the exceptionally high solar activity being a contributing factor to the losses?
There was a guy, I can't remember his name, back around CCD, when everybody's blaming CCD on windmills and the HARP project and everything else, cell phones. Mm-hmm. There was one guy who was looking at the solar activity. I looked at the data that he had, and I didn't find it convincing at that time. If it were solar activity, that would not explain why Bruce's hives and Greg's neighbor's hives are not having any problems.
Yeah.
Okay. So, I mean, if it's solar activity, it would be supposed to be uniform.
Right. Yeah.
That is interesting. Yeah.
Awesome. Well, Raina, we appreciate you jumping on.
So,
absolutely. Yes, sir. It was a lot of food for thought. I haven't put a lot of mental effort into the because my bees have done okay. I haven't thought a lot about it, but I'm thinking more about it now. And you brought up some really good points, and we addressed most of what I've heard out there, and even some of it in more depth than what I've heard. So I really appreciate you coming on. It's been a pleasure meeting you. And
Project 8% is 8% on the 28th is going to have an open meeting. Anybody can join to find out the updates from the USDA. So I would, if you're curious, I'll be there. I'll be listening for sure to see what they found.
Okay. I'll have to get the link for that and make sure that I tune in.
Yep.
Awesome.
All right. Look at everybody.
Thanks, Randy.
I hope your bees stay healthy.
Yeah.
We hope so too. Thanks.
All right. Take care.
All right, guys. You too, man. Bye-bye.
There we go.
Wow. How was that, guys? That was phenomenal. Whew. A lot of food for thought. That time went by so fast. Yeah.
That was so hard to be in the truck driving and listen to the conversation and without being able to jump in. But, if I, listening to what Randy had to say and some of the folks that you really trust in the industry, like there is no easy path forward. And we've got, some speculations that it might be this or it might be that. And that's the real, the fact that matters. So many folks are losing hives. Folks who catch swarms are losing hives. Colonies that are specific genetic lines and have been for many, many years, those are going. It's not isolated to package bees or nukes or feral bees or it is so extremely widespread. We're seeing it go through Caucasians and Carnies and Italians and bees with VSH attribute. It's going through everything. No matter, there's no respecter to genetic line, attributes, or origin, it seems. And it would be easy to just say, oh, it's this, and that's easy to scapegoat it to something that we don't agree with. But as more folks step up and say, hey, here's what I'm seeing, it seems as though it was just a year of wild strangeness just across the board. Yeah. And I think those we talked about today, those were the stressors and the catalysts of maybe an underlying thing going on. But we covered, there was to listen back to that, we just tackled and hinted at things that we've been hinting and alluding to for several years, but no one still has any answers. But to hear Randy confirm and say it is very highly likely that there is some new extra virulent thing going on, which is why maybe you see it in colonies with no mites and versus colonies with a lot of mites. Things that that leaky boat syndrome. You know, it's all these things we have to kind of think. And what's challenging about that is until we know what it is that this is, it's really hard to chart a course moving forward, right? Because if we just do all the things that we've done for so many years, right? Keep the mites back, keep them fed, keep the brood clean and healthy, get them to the right size for winter, and we do that again, like there's nothing to say we won't see ourselves here again, even if we think we do all the things right.
Yeah, Greg, one thing I think that he said that I think he was addressing a question I put out or whatever he said was he talked about the control group, and we have to be careful when we so many people, you know, you got all your colonies do well, but you treat them all the same way. So do you know that what you did is what helped those bees, or is it just they happen to do well? So you need to have some that you treat with you treat a certain way, and others that you don't, and compare, and then you'll be able to find out better. It's been interesting. I've kind of watched a little bit of what Ian's been doing up in Canada. He does some of that where he'll treat certain bees a certain way and others not, and kind of compare. Like he did that with his formic test last year. He kind of had a plan with that to kind of compare how it went for him. And so, you know, when I see all the time, I see many times in social media groups, well, obviously it's this, or obviously it's that, or if you'll just do this, your bees are going to be fine. And, but I guess the question to ask is, how did the control group do? You know, you got to compare them and see, because if you have 12 colonies and you treat 6 of them with whatever X product and the other you don't, do they look exactly the same after, or is there a definite improvement in the 6 that you treated or you fed a certain product or whatever? I think that's a really wise thing. And I don't think there's anything wrong. Like, for example, I use, you know, this year I used Hive Alive on everything, and my colonies look better than they ever have. But is that directly attributable to Hive Alive? I don't know, because I didn't have a control group. And so, you know, maybe it is, maybe it's not. And there's nothing wrong with me doing that. But for me to say that the Hive Alive is why my bees are better, you know, I don't know for sure, because I didn't do a control group. So we need to be careful, I think. Maybe that's words of wisdom to me. I haven't thought about that in that direct sense, that to make claims about a product that you've used on everything, you got to be careful about that. So that's just the thought I had on that. And I try to kind of clarify when I say stuff. My bees look great this year. I used it. I believe in it, but that doesn't necessarily mean that, hey, that's the only thing that's helping my bees look better or worse, whatever it is, whatever the case may be. If you treat them all the same, if you have huge losses, if they all look worse, is it because of what you did, or is it because, you know, just, I don't know. That was, to me, that is one of he made so many good points and so many great things to think about. But that's one point that I think that all of us could take as words of wisdom, to be careful before we say you need to do this, you need to do that, or this is for sure what works. Unless you kind of compare different things. I think that's a great thing to do. So.
I tell you, I am going to go over and so I went and checked my colonies today, and I thought I was at 3 with 1 that looked bad, but here I'm at 4 with 1 that looks just extremely bad. It's literally like the size of my fist, the cluster. And I pretty much know, and we're right now, Greg, I know you've been looking at the weather, and we're starting this pattern right here in Ohio. You notice that? You know, today we were 47.
Final nail. Yeah.
And this weekend we're going to have a low of like 17. So it's like, well, here we go. We're going to start having the 30 and 40 degree temperature swings. So that little colony over there, you know, I was telling my mom, I popped in to say hi to my mom after I was done, and she said, "Can't you do anything? Put a heat light or something?" I said, "They're dead." You know, it's just, you know, they're pretty much dead. So, Galen,
one of the things that I wanted to bring up, and we didn't have time, but you know, I'm going to try, and I would encourage everybody. You know, it's going to be very difficult or impossible to get to do this if you wait too much longer. But I would encourage everybody to take as much time as you can to start cruising through mentally through your season and start writing down all the things that now when you look back on it seemed out of place or seemed different or seemed like that is non-typical. Because I think what you might find, we might start to connect some of the dots. But one of the things that I do remember everybody saying that was not typical is, at least in our area, when that drought was so bad, you know what we were finding inside of our colonies? We were finding bald-faced hornets. We were finding all these different wasps and stressors that we never see inside of there, right? It wasn't just the honeybees, guys, that were stressed out this year. It was everything else too. And I think that plays into what we're seeing with the honeybees. And, you know, and maybe I don't want to jump to conclusions here, but based on what Randy did say, do you remember a few years ago we had Humberto on, and he was talking about the fact that hive beetles can pass along deformed wing virus, but is the variant hefty enough to cause a problem with the honeybees? They were still researching it because it was a potential. So what is the possibility that the bald-faced hornet or the yellow jacket or the red wasp or any of the other things could be transmitting a pathogen?
Right.
Is it possible? Maybe. But are these things just more stressors? I don't know, but we start doing all of those things, not seeing anything on goldenrod, nothing on aster. Everybody, even inside of the hives, looking for anything and everything to eat. Extreme drought.
Yeah.
All the things that we've talked about before, we're seeing all the, it's just, I'm afraid there are so many. We talk about this a lot when we are doing presentations, especially on getting things off to a good start. And we tell a story as we're trying to get our feet underneath of us, we're cherry picking all the things that sound good and look good and we feel like it's going to be good for us to do with our beekeeping. The next thing you know, you've got 14 different approaches that you've cherry picked all the stuff from to put together your management plan, and then something goes wrong and you can't sort out what it was because you took this approach with that, and you make so now this year we've got so many extreme variables all at once. We're leaning on immunologists and all these different things to get to the bottom of that. Like, what if the real problem's outside of that scope? We still may never know exactly what we're dealing with. And that's hard to accept.
The one thing that I had never even thought of is when Randy mentioned colostrum. And if that truly is a factor in the entire equation, okay? So you take that, and then last year we had all of the rains and everything, and then you had the drought, and you had all of these natural sources that were just producing these poor foods for the bees. So you're having poor nutrition fed to those bees that should be going through the winter. And then any other factors. You know, I think it truly is, you know, like I said, it's just this perfect storm of events, that leaky boat that was a
huge point that he made, though, because if you think about it, when we had that much of a drought and there was no fall pollen for those bees to build up on, the nurse bees that are setting up the winter bees to be successful probably didn't have the nutrition that they needed. And I can't say every, but I can tell you without a shadow of a doubt, when we get into late July and August and September, I am not willy-nilly putting 1-pounder pollen patties on every colonies just in case, because the great fear in my neck of the woods is hive beetles. So we're not going to put anything on there and hive beetles started to creep up at that time too. So now in hindsight, it's like, okay, well, should I have done that? Should I have risked the hive beetles? Would that have helped? What was the right? But it's you end up with more questions than answers, you know.
But it was definitely just an incredible thing, and I would encourage anyone who is watching now or just coming into the stream now that, I mean, Randy just got off a couple minutes ago, but go back and watch if you didn't see the first part of this. It is just, Randy just had so much incredible information and insights on things that that many of us probably never thought of. Greg, you mentioned the word cherry picking earlier. So many of us, and I'm guilty of this for sure, we also cherry pick sometimes the best of what we do as beekeepers. And we sometimes, I think, will tend to not focus on the negative things that happen. And so it's important to look at the whole picture. Whenever, Greg, especially with your business, I know you have to look at the good things and the bad things. You can't just look at the profits. You have to look at the things you did that weren't successful and take a real look at it. Every dime, every penny, every dollar has to have a name. And if something doesn't work, you have to pivot. I think it's the same way with beekeeping. You can't just look at the successes, or you can't just look at you've got to look at the whole picture. And then if you continue to do that and look at the positive and the negative, and you continue to move forward with the positive aspect, you'll become more successful. If you have the ability to pivot and not just be in one mindset, you're going to be more successful. Mm-hmm. And that's going to happen. Like Bob said, I think Randy alluded to this too a little bit. There have been waves of this kind of thing that have happened before, and it happens in cycles. And I know Bob talked about that with you, Greg, that every few years there is a die off of bees. There is a thing that happens, almost like a reset. Mm-hmm. And everybody gets really upset and worried about it. And it is a concern, but if you can just be, as Bob says, tenacious, you can overcome. And like Randy said, those who are willing to adapt and who are strong enough and willing to do that, he used the word opportunity. And that applies in so many aspects of our lives. If a negative thing happens, if it's a difficult time for you, there's opportunity on the other side if you're just willing to be tenacious and stick with it and move forward. Okay. If it's what you want to do. Now, if you, like he mentioned, some of the older beekeepers that just are going to get out, or people that just don't want to deal with it anymore, and that's perfectly fine if it's not what you want to do. But if it's something you love and it's something that you're determined to do, don't give up. Keep moving forward and be tenacious, and you will be able to have many opportunities coming out on the other side. So I love that he said that. I like the way Bob mentioned that as well in your interview with him, Greg. Mm-hmm. This is a difficult time for many people.
Yeah.
And I can't imagine if I was down to, Greg, in that situation where you lost 98 of 100 colonies. I can't imagine if I had 144 colonies last summer and now I had 4. I mean, I can't even fathom that, losing that many colonies, so
yeah.
But I would hope that I would be able to figure out a way to kind of start over and rebuild and get back up to where I want to be. I haven't experienced that, but if I did, that would be extremely difficult. And I totally understand people that would say, you know what, I'm just going to go find something else to do. It makes total sense because there's a lot of work that goes into these bees. But yeah. I did like the word that Randy used where he said opportunity. That means a lot to me. And I love that he said that. So, man, this has been a tremendous chat, guys. Don't you think? It's we've been going for a while.
I know it's been a good one. I mean, we're what, hour and 50 minutes?
Yeah, so we're almost at that 2 hour mark. So
there's still 240 people.
Yeah, we appreciate everyone being on here too.
Yeah.
So Brian, what you got coming up in the next little while?
Well, before I get to that, I do want to say, Mr. Bruce, thank you. Pat, thank you. Steve, thank you. Bees in the Abyss. I like that name. Bees
That's pretty cool.
The Abyss.
That's what this year feels like, doesn't it?
I know. Licking Mo Honey. Look at that. Thank you much. And Galen, that is awesome. Thank you. Thank you so much. So just want to thank everybody for the support, always tuning in. So just so awesome. But you know what I have coming up? So let's see here. I guess I need to get back and watch a replay. Yes, Terry, you do. But I went over today and it was raining. The weather up here it never just ceases to amaze me. I'll just say that.
Relentless this year, huh? Relentless.
Yeah. Living in Ohio, it is what it is. So I'll leave it at that. But I went over and it wasn't really that bad of a downpour. Everybody here has gone through and you've done some work in a drizzling rain. So I wanted to peek and see how the colonies were. The temperature, like I said, we were close to 50. It was about 48 degrees. And there are 3 colonies that I have that are, I feel, really comfortable in that they will live. They're just, I'm talking on the top boards and like on the insulated cover when I peeked it, propped it open. I mean, just decent sized cluster of bees. So those three will probably be my surviving stock that I'm going to have a plan to utilize those as a foundation. But like next week here, our temp still, I can't even think about splits. I can't think about anything that you're doing right now, Bruce, because our weather just doesn't cooperate up here. So, but I've been in like equipment prep mode. So I've been scraping frames. I've been going through my frames, and if I see frames that have just that exceptionally old comb, I'm scraping it. So I'm getting all of that type of stuff done. And I do need to look at like bottom boards and all that type of stuff here. That way, the next time I go down to Greg's, that I can just get everything that I need and have everything set. So I know packages will be coming up in month and a half. So I've got to have everything set, and I don't want to start the season with everything that has taken place. I don't want to start the season backed up in a corner. I want to have everything situated, everything ready, so that when I do need to work the bees, well, I'm not having to spend an hour in my honey room doing this and that and whatever, before I get to the bees. So I want to get everything set so that I can really focus, really concentrate on everything that I'm doing with them this year. So, but that's about it. I will have a video. I did film a video today. I did show my one colony that was dead, just so people could see. I've had people saying on the social medias and on the videos saying, oh, they probably didn't have food. And I literally held up one frame and there were dead bees like 2 cells away from food. But with the weather and how it's been, the cluster wasn't like a really tight knit cluster. They were expanded out some. So, I truly, I'm just blown away with that. So, and who knows? So, but I could keep going, and I'm almost turning into like Greg Burns mode where I'm doing short story long here. So, but I'm going to pass the. I know.
Where's mine?
Got to dial it in. See, dial it in. That's right.
So, you got to remember that technique. I didn't want. Yeah. Well, down here in Alabama, I am in full blown split mode. It's just crazy. I've gotten queens from Zach Heinzman in the last 2 weekends on Valentine's Day, February 14th. You might see Jeremy in here. He spells his name Jeromy. He's the young man that's helped me out. He helped me out 2 weeks ago on Friday the 14th. And the video just dropped today of those splits. It was a really good day. We had a lot of success. We split a bunch of colonies. We queened, I think, just a couple. We didn't requeen many of those colonies. They were actually a little calmer this time, Greg. The ones that I thought were mean, they weren't as bad. And so we had a tremendous day. We had a lot of splits, and we had every single slot, I call them, on those pallets is full in both Slocum and Appam farms, except for one on the pallet where the Appam hives are. We're missing a hive there. So we got 51 in Slocum and Appam farms, and 32, I believe, out in Slocum. And then this past weekend, my goal was to help tame some of the spicy ones that I have still up in what I call my northern route. And that was quite the adventure, because on Friday it was actually pretty chilly to be working bees. And I don't know if they were just cold or what. They were fired up, but it wasn't as bad as I thought it was going to be. But I actually, I did eliminate some queens. We made some splits, and the next day, Saturday, I went in and I finished up the job, and I put the queens in the colonies. And towards the end of the day, it was getting cold, and some of the queens almost to stop moving. So I was worried about that. I had them out probably, I didn't realize how cold it was getting. But I went back and checked when I thought was a goner. I thought she was dead. I checked in that colony yesterday, and she was moving around. They hadn't released her yet, but they were about to release her. So they revived her somehow. So we'll see how that goes. I think if these queens can get released, if they don't get killed by the bees, they can get released and take over these colonies, we're going to have a much better attitude in the northern route as well, up around the farm, Midland City, Cell Tower, bee yards. I did shoot some video on that as well, but I haven't got it edged yet. I just got the other one put up today. So But it's been exciting. It's full speed ahead. This coming Friday and Saturday, my plan is to go down to that apple and bee yard, Greg, where we worked that day in that video, and I made really strong splits on a lot of those, and I'm just going to take a bunch of your boxes down there, the EnduraHive boxes, and we're going to just, the ones where the 10 frames, we're going to just put them right into the EnduraHive boxes. They're in nukes now, most of them, so we'll just move them right into 10 frame boxes, the EnduraHive, and really going to try to get that Apple Farm bee yards. The first one I'm going to try to transform into the EnduraHive, Propola, Bruce's Bees logo.
Mm-hmm.
There's going to be some other boxes out there, but most of them are probably going to go initially in that bee yard because that's the one that's most visible, and right now it's my favorite bee yard, and so I'm going to really try to kind of convert that one first. I'll have some mixed in other places, but that's the one I'm going to try to convert the most quickly here initially. And so, but that's what's going on here. I always get frustrated because I can't produce more videos and get more stuff out to you, but I'm doing the best I can. I also get frustrated because it takes me a couple of weeks sometimes to get it out, but I'm just trying to get it done, guys. And I appreciate everyone in the chat here who's here tonight and who supports the channel and all the positive comments that I get and folks that are so supportive. I appreciate every single one of those. But that's what's happening down here at Bruce's Bees.
Awesome.
Greg, why don't you take it away for us?
I want to thank everybody for the question. Brian Bennett, is there a long wait on getting boxes? No, not typically. It depends if we end up needing to do custom boxes. If you want boxes hot branded or anything special or different, then there's going to be a little extra lead time on that. But if you're looking for equipment in bulk by the skid, no problem. Be sure to go to our website, naturesimagefarm.com. Click on the About Us and shoot us a message. We have been traveling a lot in the last couple months, and so we're working on getting caught up on all the emails and things like that. But yeah, that's it's been a lot of fun the last couple months, especially last month, visiting with beekeepers like Bruce, going down and seeing Lamar down in South Carolina and visiting with him and his family. And like one man countenance is to another, iron sharpens iron. It's been such an incredible opportunity to learn from beekeepers, spend some time with them, hear their ins and their outs and their stories, their ups and their downs, the hard lessons learned. That kind of brings us to what we're looking at right now. It's thank God it's such a blessing that we had more than 100 colonies. Of that yard of 100 where 98 of them died, I look at it a different way. I look at it as a silver lining. There's 2 colonies there that are tremendous lighthouses in that particular yard, that something didn't faze them, something didn't bother them, and somehow they stood strong through all of this. So what that does for me is automatically says these are good ones to take a look at for breeding. There's something special there. So I always, you're never going to forget when you lose 90 out of 100 in one of your yards. But there's always, it does no good to sulk. It does no good to get caught up in the woe is me and I can't believe this is not going my way. And now what? It does zero good. Everything in life is just an experience. Sometimes it feels good. Sometimes it's bad. But it's an experience, and what are we going to learn from it? How are we going to move forward? There's no time to sit and sulk. What are we going to do to keep things moving forward? That's the name of the game. And no matter which way you cut it, we're all bee farmers, and no matter what the crop is, if it was easy, everybody would be doing it. I think we're turning the page of a new chapter in beekeeping. I think the beekeeping in the future is going to look a lot different than the beekeeping of the past. That's both a little frightening, but also very exciting, because I think through adversity, there's great change and great hope, and there's a great future ahead of us. If we are in the bottom of the barrel, of the bottom of the valley, of the valleys and mountains of beekeeping, guess what the great news is? The darkest hour is just before dawn, right? Tomorrow's a new day. This is going to be a new season for everybody. It's onward, it's upward. Let's dig in. Let's do the best we can. Let's support each other. Let's lift each other up. And we're just going to keep moving on, and we're going to keep doing it together. So everyone go ahead, Brian.
When you say that, I periodically I think of something that Tara, my wife, and I did during our wedding ceremony. We did the cord of 3 strands, and well, there's 3 of us, so I definitely know that I'm stronger in my beekeeping because of the 2 of you.
Oh.
Also stronger with this.
Mm.
So, but today when I was sitting there talking, filming, I did a little video. I was sitting there and it just popped in my head, and just with everything going on and just all the bickering and this and that and whatever, we all, regardless of what scale you're at, we're all having an issue. And if we all, being a hobbyist, being a sideliner, being a commercial guy, if we all work together, we are stronger. And that's what we have to do. We have to all work together. We have to all be kind to each other. We're all going through challenging times. I know John is on here. He's lost 43 colonies. There's a guy in Rob's study that's lost 680 colonies. So it's challenging times, but working together, we're stronger and we'll do better,
Yeah, that's exactly it. You never want to say, it's easy to say count your blessings. It's easy to say no matter how bad you have it, somebody else has it worse. Those aren't things that you necessarily want to lean on to for hope and for joy. But the fact of the matter is, if we all look back at any point in our life, there has been ups and there has been downs. And I think as we are going through and we are suffering through these moments and we are suffering through what to do next and all these different things, for me, I lean very heavily on my faith and not lean on it as a crutch, but utilize it for the power that is there. And it's been a reminder, lately, that all things work to the glory of God for those who love the Lord. God we're everything is going to be fine. We're just going to keep putting one foot in front of the other. We do not have to know all the answers. All we have to do is know the one who does. And when God's in control, God's on the throne, none of this, we're going to be fine. We're going to work through it. We're going to figure it out. And when we put God first and then our family and then our bees, all is well. And so I want to encourage folks and just remind you that the darkest hour is just before dawn. We're going to pull through. We're going to push through. It's going to be a different year coming all together. And it's going to be really, really exciting. So Tom Nolan, I love these AA meetings. Us APIS addicts need the mutual support. Amen. That's you know what, that 100%. Absolutely. Guys, next week is on Bruce's channel.
Yep.
Okay, Bruce's channel, Wednesday, 8 p.m. You don't want to miss it. It seems like every week there is just so many new developments. And you would think at some point this is getting old talking about it, but there is always something different and a new angle and something else that's popping up that we're learning. And that's going to be the name of this game, is we're just going to keep working through it. But thanks so much for spending time with your favorite Wednesday night bee chat. We're your favorite because we're the only one, I guess. But hey, we'll take it. But anyways, we thank you so much for spending your time with us. Keep your eye on the prize. Darkest hour is just before dawn. And we always want to remind you, as always, to be the lighthouse and be the change you want to see in this world. We'll see you next time. Thanks for watching.